Hello everyone, welcome to my channel. In today's topic is going to be about cell part 3 for your NEET preparation. And in case you have missed any previous episodes, I will link it in the description box so that you can follow up in this part 3. So in the previous class, I've covered about fluid mosaic model of the plasma membrane. And also if you're interested in any other topics in science or NEET, you can always leave it in the comment section, a message saying that this is the interested topic and I will make a video on it. And if you're interested in the notes of this presentation, you can also leave a message in the comments below. And if in case you have not subscribed yet to my channel, please do subscribe so that you will receive notifications whenever I make some video. So let's get started. In today's topic, I'm going to cover mostly about cell wall, endoplasmic reticulum, and Golgi complex. All right, so let's get started. So beginning with cell wall, I hope we all are aware that only plants and algae have cell wall. Only plants have cell wall and algae have cell wall. Animals and we do not have cell wall. All right. So if this is the cell, this is the cell membrane, this is the cell wall. All right. We have the plasma membrane, but we do not have the cell wall. Okay. So cell wall is a non-rigid structure that forms an outer covering for plasma membrane of fungi and plants. So this layer is the plasma membrane, right? So cell wall forms the outermost covering for the plasma membrane. And let me tell you something. What are the functions of the cell wall? So first is it gives shape to the cell, but most importantly, it protects the cell from it protects the cell from mechanical damage and infection. So if there is any damage that is going to be done or an infection that's going to cause harm to the cell, this layer protects it. Okay. So what does it do? It protects the cell from mechanical damage and infection, and it also helps in cell to cell interaction. Okay. So it allows good things to pass through, like let it be some water or food. Transport of food occurs through phloem and water occurs through xylem. Food and minerals occur through phloem. We'll be talking about how it is used in higher classes, but for now, remember that it helps in cell to cell interaction, but it does not allow any undesirable things to pass through. Okay, so how is the cell membrane in algae different from that of the plants? Okay, so algae have cell wall that has cellulose. This is a neat question. So this is a neat question which has been asked. So cellulose, galactans, mannans, and minerals like calcium carbonate, whereas in plants you have cellulose, hemicellulose, pectins, and proteins. All right. So here you can see in both of them only cellulose is present, rest are different. So remember in algae it is cellulose, galactans, mannans, and calcium carbonate, whereas in plants it is cellulose, hemicellulose, pectins, and proteins. Okay. Now, so what are the layers? Okay. Actually, there is primary cell wall. The cell wall of the young plant will have primary cell wall, and after it matures, secondary cell wall will be formed. In some you can see tertiary cell wall also. Okay, and you can see middle lamella. Let me show you the picture. So if this is the primary cell wall, it will mature and later form secondary cell wall. So this is the primary cell wall and it has this middle lamella. So basically what does this middle lamella does is, so if this is the middle lamella, if these are like four cells, okay, Plant cell means doesn't mean whole plant has one cell, right? There are a lot of cells. So if this is the cell membrane and this is the primary cell wall, so this middle lamella is something like this. So it will help in connections. Okay, it will help to transport things from this cell to this cell and this cell to this cell. Okay, so middle layer is mainly composed of calcium pectate. So what does the middle layer contain? Middle layer consists of calcium pectate. Okay, it mainly glues the neighboring cells together. Okay, the cell wall and middle lamellae may be transferred by plasmodesmata. So there might be some branches, okay, which connect cytoplasm of both cells. So these branches that are connecting the cytoplasm of both cells are called as plasmodesmata. So this is middle lamellae, but this cytoplasm and this cytoplasm is being connected by some branches, and those branches are called as plasmodesmata. All right. Now, cell wall of a young plant cell is capable of growth. Okay, so they're talking about the primary cell wall, and after it matures, secondary cell wall is formed. Okay, and middle layer is formed. So what is the middle layer formed of? It is made up of calcium pectate, and the function is to glue two cells together, and there might be transverse branching that is called as plasmodesmata, which connects cytoplasm of neighboring cells. Now, the second subtopic which we are going to study right now is about endomembrane system. All right, so in which we'll be studying about endoplasmic reticulum. So electronic studies of eukaryotic cells reveal the presence of network of reticulum of tiny tubular structures that are scattered in cytoplasm, and they are called as endoplasmic reticulum. So let me tell you something. Endoplasmic reticulum, here you can see there are two kinds here: rough endoplasmic reticulum and smooth endoplasmic reticulum. So rough endoplasmic reticulum has spikes. That's how it looks from the picture. Whereas smooth endoplasmic reticulum is pretty much smooth. So these spikes are nothing but ribosomes which help in protein synthesis. So if endoplasmic reticulum has the ribosomes, it is called as rough endoplasmic reticulum. And if the ribosomes are absent, then it is called smooth endoplasmic reticulum. Okay. And let me tell you something. The intracellular space is divided into two components. Okay. So one is luminal, that is inside the endoplasmic reticulum, and extraluminal is in cytoplasm. Okay, so how did it divide? It has divided into two things. One is luminal and extraluminal. All right, so luminal is inside the endoplasmic reticulum and extraluminal is in the cytoplasm. Now let us talk about rough endoplasmic reticulum and smooth endoplasmic reticulum in detail. So as I told you, rough endoplasmic reticulum has ribosomes on their surface. And the main function of this ribosomes is it helps in protein synthesis. All right, it helps in protein synthesis and secretion. Okay, and they are extensive and continuous with the outer membrane. Now coming to smooth endoplasmic reticulum. So they do not have any ribosomes on them. That's why it is called smooth endoplasmic reticulum. But what is it important for? It is a major site for synthesis of lipids. It helps in lipid synthesis. All right. In animal cells, lipids like steroidal hormones are synthesized in smooth endoplasmic reticulum. So what does the smooth endoplasmic reticulum give? It gives steroidal hormones. All right. So what did we study here? We studied that in rough endoplasmic reticulum, it is mainly for protein synthesis and secretion. They are extensive and continuous with the cell membrane. And smooth endoplasmic reticulum are mainly the site for lipid synthesis, and they also help in synthesis of steroidal hormones. All right. Now coming to Golgi apparatus, this is how Golgi apparatus look like. Okay. So let me show you here in the animal cell. This is rough endoplasmic reticulum, and you can see smooth endoplasmic reticulum over here. And if you can see, Golgi apparatus is in close proximity with the rough and smooth endoplasmic reticulum. All right. So who observed Golgi apparatus? So it was Camillo Golgi. It is his name, and he has given the term Golgi apparatus in 1989. And he observed the structure near the nucleus. And they consist of flat disc-shaped cells. So they have like flat disc-shaped cells. They are called as cisternae, and they are 0.5 to 1.0 micrometer. Like here you can see flat cisternae. Like you can see these, right? So those are flat cisternae. And they are stacked parallel to each other. Here you can see how they are arranged parallelly to each other. Okay. Cisternae are concentrically arranged with convex cis forming surface and concave trans maturing surface. So there are two surfaces, one is cis surface and one is trans surface. Okay, cis surface is considered to be the forming phase, and trans is maturing phase. So it forms and matures like this. If you see in this way, this is cis and this is trans, because it's forming and then it's maturing. All right. So what is the function of this is it packages, like, it helps in packaging the material to be developed either to intracellular targets inside the cell wherever it needs the material to be delivered. It helps in packaging and delivering it, or if anything needs to be secreted outside the cell. So mainly you can remember it as packing and delivering help organelle. Okay. Materials to be packed in the form of vesicles. So they help in the formation of vesicles from the ER, fuse with cis phase of Golgi apparatus and moves towards the maturing phase. Okay. So remember, so from here, from the endoplasmic reticulum and smooth endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum, this Golgi apparatus helps in formation of vesicles. Okay, from the ER. So, from ER, it is taking and packing it in the form of vesicle and giving it inside the cell or outside the cell. That is the reason why they both are in close proximity. A number of proteins synthesized by ribosome on endoplasmic reticulum are modified in cisternae before they are released from its trans surface. So, before they enter the trans surface, the number is already modified. Okay, the number of proteins is already modified on the cis phase. So, let me show you. So, number of proteins synthesized by the ribosomes from the ER are modified in the cisternae before they are released into the trans phase. Okay, and Golgi apparatus is important site for the formation of glycoproteins and glycolipids. So I hope you have understood about these three cell organelles, that is the cell wall, endoplasmic reticulum, and Golgi apparatus. So I have already made 3 videos. One video is about basics of cell, and then I give a detailed explanation of each and every organelle. All right. So we are currently in the part 3, and tomorrow I'll be talking about part 4 of the cell. But let us have a quick review of what we have learned today before I end the session. All right. So what did we learn about the cell wall? Cell wall is nothing but it is a non-living rigid structure. It can be seen in the algae and plants, and it is outside the plasma membrane. It gives protection, it gives shape to the cell, and also it helps it from unwanted things, and it helps in communication from one cell to another. And there is a difference in composition of algae and plants cell wall. In algae, the cell wall is made up of cellulose, galactans, mannans, and calcium carbonate, whereas in plants it is made up of cellulose, hemicellulose, pectins, and proteins. All right. Now there are like primary cell wall and secondary cell wall. First, the primary cell wall will mature, will form, and it will mature, and then the secondary cell wall is being formed. And then you have a middle layer, and that is middle lamella, which mainly has calcium pectate. And what does it do is it helps in gluing the neighboring cells together. There might be branching where you can see like transverse lines. They are nothing but plasmodesmata which connect the cytoplasm of the neighboring cells. Now coming to endoplasmic reticulum. So endoplasmic reticulum are tiny tubular structures. They are scattered in the cytoplasm and they divide the intracellular space into two components. One is inside the ER, it is called as luminal. Outside, it is called as extraluminal. Luminal occurs inside the endoplasmic reticulum and extraluminal occurs in the cytoplasm. And endoplasmic reticulum is of two types. One is rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum has spikes like structure that is nothing but the ribosomes that help in protein synthesis, and smooth endoplasmic reticulum lacks those ribosomes. So that's why it's called smooth endoplasmic reticulum. And rough endoplasmic reticulum mainly helps in protein synthesis and secretion, and smooth endoplasmic reticulum helps in lipid synthesis and steroidal hormones. Now coming to Golgi apparatus, it is a cell organelle near the nucleus. It was coined as Golgi apparatus by Camillo Golgi in 1898. They have flat disc-like shaped cisternae of zero point 0.5 to 1 micrometer. And let me show you here. These are the cisternae and these are stacked parallelly with each other. All right, they have cis, that is forming phase, and they have maturing phase, that is trans. So cis is the forming phase and trans is the maturing phase. And they mainly help in packaging and delivering the content in the form of vesicles inside the cell or outside the cell wherever it is needed. So how do they form? These vesicles are from the endoplasmic reticulum, and they form vesicles and then they help in this transport. That's the reason why they are in close proximity near the endoplasmic reticulum. Now, the number of proteins that are synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from the trans surface. And Golgi apparatus is important in the synthesis of glycoprotein and glycolipids. So today we have covered cell wall, we have covered endoplasmic reticulum, and we have also covered about Golgi apparatus. We'll be talking about plastids and lysosomes, nucleus, and all the other cell organelles in the next class. But in case you have any questions regarding NEET or any science topic in general, leave a comment below and then I will be very glad to help you out. All right. So thanks for watching my channel and stay tuned to my channel for more updates. And also please do subscribe. Thank you and thanks for watching.